Can somebody confirm you can hear me?
I can hear you.
Thank you. Sorry for being a little bit late, but they've changed the way that I log in. And now I have to verify through an email. And the first email didn't verify it. I'm not sure why. And so I had to request they send another email. But we're in now, so hopefully everything will be good. Before we begin, are there any questions? Sorry, I've got a frog in my throat.
Yeah, about the infectious disease project. I'm just getting close to being done with it, and I'm wondering if there's a word count range that you're looking for, or it's just like it can't be more than 5 pages, but how
Uh,
much less than 5 pages it can be. I'm just kind of wondering.
it should be around 5 pages. If it's more than 6 pages, you'll get a penalty. That's not counting any pictures or the references. Okay. If it's less than, let's see, 4 pages, you'll also get a penalty. So something
For.
between 4 and 6 pages.
All
And
right.
I don't have a word count, so I don't check that.
Okay. All right. Thank you.
All right. Let's see. Today is February 19th. And we're supposed to be starting chapter 6. I think we need to finish up chapter 5. Let me check and see. I don't have a 2 here, but let me look. Oh, that's not the right place. I think we've gone past this. Let me see if there's another 2 here. Ah, there we go. Photosynthesis. That's where I thought we were. All right. Let me share my screen. Let me state I have done some grading. I believe on the Monday and Wednesday I've got grade. Maybe I should look. I've got lab 6 graded. I think I graded some other things, but I don't remember what it was. Well, that's not going to help. I'm going to the grade book. That's why I'm not sharing my screen. I see I have a lot of emails again. I just sent a whole bunch of emails out on Sunday. I think I did it on Saturday as well. I've not graded extra credit study 6. I've not done the lectures. I'm sorry about that. I'll try and get to that. But yeah, worksheet lab 6 is graded for you guys. Oh, and the unknown notes was graded or updated on Friday or Saturday. I did not look at the unknown notes on Sunday. I did promise that I will not be looking at the unknown notes on Sunday. All right, any questions about the grading? Let me get out of the grade book and then I'll share my screen. I think I've got the syllabus, the new syllabus with the new schedule up online. Let me check on that now and I will share my screen while I'm checking. Come on. All right, you should be seeing my screen now. And where's my syllabus? Yep, back one page. Well, that's not right. Oh, here it is. So it was page 11. I just didn't see it. Yeah, the reviews on the lab now. So yeah, this is the new schedule. It's on the syllabus now. I'm not sure when I made that available, but it's now available to you. All right, let's take a look at the syllabus. And today is February 19th. We're supposed to be on chapter 6, but we've got to finish chapter 5. Let me warn you that quiz 3 is this week, Wednesday, and it's going to be covering lab 5 and 6 in the lab, and chapter 4 and the end of chapter 3 in the lecture. Please note that your infectious disease paper is due this Saturday. So please get it in by 11:59 p.m. this Saturday. We do have a lab today. I should say this week. I'm not sure why, but I've got the first day. We're not having the lab. You're just working on the unknown project. And then the Wednesday, we'll have lab 8. Any questions about any of that? All right. If not, let's go to chapter 5. So I need to talk about photosynthesis. Photosynthesis is the conversion of light energy into chemical energy. Specifically, most people think of it as the conversion of energy in light to the energy in sugar. However, sugar may not be the product made in photosynthesis, although you've never probably been told that yet. Chemical energy may be used to convert atmospheric CO2 into glucose, and this is called carbon fixation. But it may, instead of making glucose, make ATP, and I'll talk about that. Photosynthesis is performed in photosynthetic organisms like green plants. Everybody knows that. Green algae. But then there's green bacteria too, such as the cyanobacteria. And there are photosynthetic organisms which are not green. They have a different pigment that is not green. Like red algae, I'm sure everybody's heard of that, is photosynthetic. And it does not have chlorophyll, so it's not green. It has another pigment. I don't know what it is, but it's another pigment. A photosynthesis is a mechanism for synthesizing organic compounds, and this is very important for animals and microbes that feed on matter from other organisms. Most niches on the planet Earth depend on the photosynthetic organisms to produce glucose for them. The only exception is the deep sea vents and maybe some deep, what do you call it? Caves, usually water caves, where there may be chemosynthetic bacteria supplying the energy for the niche. But in all other niches, photosynthesis is required. Let me move on. So photosynthesis only happens in photosynthetic organisms, and they contain a light trapping pigment. One of them is chlorophyll. That's the light trapping pigment everyone is familiar with. It's in green plants. There are other photosynthetic pigments, and if you don't know, most green plants have a yellow pigment, and you can see the yellow pigment in the fall when the leaves turn yellow. But most of the time, the leaves have their primary, meaning the majority of their pigment being chlorophyll, and so the leaves are green. But in the fall, the chlorophyll pigment decreases, and so the yellow pigment then becomes visible. The red pigment in the leaves, just for your information, is not photosynthetic. Anyways, light energy can be converted to chemical energy, and this happens by an electron transport chain. ATP and NADPH are always generated in photosynthesis, but they may be used, meaning break down ADP and NADP, to make glucose from CO2 and water. There are two types of photosynthesis: cyclic photosynthesis and non-cyclic photosynthesis. All of you have probably been taught non-cyclic oxygenic photosynthesis, where we have 6 CO2, and you should know this reaction, plus 12 water in the presence of light produces 1 glucose, C6H12O6, 6 oxygen atoms, not atoms, molecules, and 6 water molecules. Sometimes you'll have the 6 water molecule removed on this side, and then they'll remove 6 water here to balance the equation. I prefer this equation because it has 12 oxygens coming from water, and all of those oxygens produce oxygen gas, meaning the oxygen that's produced in photosynthesis. None of this oxygen is produced from carbon dioxide. You probably have not been shown the anaerobic photosynthesis where oxygen is not produced. Instead, we produce sulfur. You don't need to know this chemical reaction for photosynthesis. However, you should know that there is another type of photosynthesis. It does not make glucose. Cyclic photosynthesis only makes ATP. In cyclic photosynthesis, ADP and phosphate are made into ATP. And if the plant or if the photosynthetic organism requires ATP, it'll engage in cyclic photosynthesis. But if the photosynthetic organism wants glucose, it'll engage in non-cyclic photosynthesis. Any question about that? And as I stated, in non-cyclic photosynthesis, we do make ATP. However, that ATP is used to make the glucose. So the ATP is not kept. Any question about any of that? All right, let me blow this up a little. This is showing you the cyclic photosynthesis, where light strikes the chlorophyll in photosystem 1, meaning that's one pigment in this system of a bunch of proteins, and the light is absorbed by an electron, causing that electron to move. And that electron moves through an electron transport chain, first to this protein in a membrane, then to that protein in a membrane, then to that protein in a membrane, and then back to this protein in the membrane. That electron actually travels back to chlorophyll, the molecule the electron came from. Why this is called cyclic photophosphorylation or cyclic photosynthesis. The electron travels in a cycle, coming back to the place for where it started from. Along the way, the electrons give this protein the energy it needs. Oops. Sorry. It gives this protein the energy it needs to transport hydrogen ions from this side of the membrane to that side of the membrane. This electron transport chain should be familiar to you because it's very similar to the electron transport chain of aerobic respiration. The hydrogen ions then create a twofold gradient on this side of the membrane compared to that side of the gradient membrane. There's more positive charges here and then more hydrogen here. That means these H+ ions want to move over here. This is potential energy. The cell uses that potential energy to make ATP. The only place the hydrogen can move through the membrane is at the ATP synthase protein, And when the hydrogen ions move through this protein, they give the protein the energy they need to phosphorylate ADP and make ATP. And this too should be familiar to you because this is the way that aerobic respiration makes ATP. Any question about cyclic photophosphorylation? All right. The last slide is showing you a summary of ATP production. The energy source can vary. The energy source for photosynthesis comes from the energy in sun, hitting the electrons, causing them to move. The energy source for organisms engaging in glucose metabolism obviously come from glucose, and the electrons come from glucose and then flow through an electron carrier chain, making ATP. And that's true for the electrons that are stripped from a inorganic chemical like iron or nitrite or nitrate. And those are in a chemosynthetic bacteria or chemosynthetic archaebacteria. The electrons move through an electron transport chain to make ATP. The final electron acceptor can vary. I'm not sure why it's not shown in here, but for cyclic photosynthesis, the electron acceptor is the same as the electron donor, the chlorophyll molecule. And like I said, I don't know why that's not shown in there because they have it right there. The electron source is varying, so the electron acceptor for photosynthesis should be in this slide, and it's not. The electron donor final electron acceptor can also vary for glucose metabolism. There's aerobic respiration where the final electron acceptor is oxygen. And anaerobic respiration, the final electron acceptor is a molecule other than oxygen, such as nitrate or sulfate. And then for fermentation, the final electron acceptor is an organic chemical compound, either pyruvate or the end product of fermentation. Any question about this? All right, that's it for chapter 5. Let's move on to chapter 6. I don't want to stop that. I'm going to stop here. Here we go. All right, this slide is showing you the major goals and a rough outline for chapter 6, microbial growth. Know the terms of chapter 6, and you really should look at the objectives because it'll have all of the terms. This is only the major terms, major goals. And then 2, understand the various requirements of cells for growth. 3, understand the phases of growth. And then 4, compare the different methods we have for measuring microbial growth. Any question about this overview? If not, let me inform you that when we're talking about microbial growth, we're referring to the increase in the number of cells, not to the size of the cells. I used to teach middle school students, and when we said they were growing, we did not mean they were reproducing. But when we're talking about bacteria cells, when we talk about them growing, this is not opening, sorry. There it is. We refer to them as reproducing.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Any questions about that? All right. So cells have requirements for growth, and this is true for microbes as well as really all other living organisms. And they have two major requirements, physical requirements and chemical requirements. Physical requirements include things like cells have to be at the correct temperature for microbes to grow. They also have to be at their correct pH, their correct osmotic pressures, their salinity. Chemical sources include things like all living cells have to have a source for carbon, a source for nitrogen, a source for sulfur, a source for phosphorus or phosphate, a source for trace elements, a source for oxygen if oxygen is required, and if oxygen is not required, you might require oxygen to be absent. Many anaerobes require oxygen to be absent. And if the cell requires it, they have to have a source of an organic growth factor. Like for humans, all of our vitamins would be an organic growth factor. And for humans to grow and reproduce, we have to have a source for vitamins. Any question about any of that? And yes, microbes have organic growth factors too. If you remember, Streptococcus pyogenes gets its organic growth factors from the red blood cells. And I think one of them is NAD plus, but I'm not going to swear to that anymore. All right, let's go on. So each species has a temperature requirement. Each species has a minimum temperature it can grow at, a maximum temperature it can grow at, and an optimum temperature that it can grow at. Microorganisms are actually classified into groups on the basis of their preferred temperature range. Let me blow this slide up. We have psychrophiles. These are microbes that grow best around 10 degrees Celsius. That is the temperature of your refrigerator. These microbes cannot grow in us. Those enzymes would disassociate, their membranes probably would disassociate as well. Psychrophiles cannot grow in humans, and they cannot directly cause humans to become sick. However, they can grow in food in the refrigerator or elsewhere, and they can cause the food to spoil, so they can indirectly cause humans to become sick, meaning from food spoilage. Any questions about the psychrophiles? The psychotrophs are organisms that grow best around room temperature. That's around 20 degrees Celsius. These are organisms that grow typically in the soil. They may be organisms in your house, like in the dust in your house, or floating around in the air. These organisms cannot grow in humans. The enzymes would fall apart or would unravel. They wouldn't be in their three-dimensional optimal shape. These organisms can cause humans to be sick indirectly, though, and that is they can cause food spoilage. Mesophiles are organisms that grow best at 37 degrees Celsius. If you don't know, that's body temperature, or 98.6 degrees Fahrenheit. All human pathogens are mesophiles. These are the organisms that cause us to become sick when an organism invades us and starts growing in us, meaning when we have an infection. Thermophiles are organisms that grow best at 60 to 62 degrees Celsius. I don't know what temperature that is, but it's fairly warm. I do know that if you ever see a chicken that's at Safeway or some other grocery store in a rotisserie going around and around, and you look at it and you say, geez, that chicken looks pretty old, it probably would be a good idea not to buy that chicken because thermophiles can grow under the heat lamps of that rotisserie. And so if the chicken looks like it's been there for a long time, the thermophiles could begin growing in it. The hypothermophiles were something that weren't even known when I took microbiology. These are organisms that grow best around 95 degrees Celsius. Recall that 100 degrees Celsius is the temperature that water boils at. Any question about any of that? These organisms do not grow in humans. Their enzymes would freeze. Their cell membranes probably would freeze as well. And so these cells cannot survive in the temperature of a human. Surprisingly, the thermophiles can make you sick by food spoilage. Surprisingly, the hypothermophiles, we don't have any evidence that they cause people to be sick. And we do know that some Mexican women or maybe Hispanic women, when they live around a volcanic vent, they'll put their corn on the cob in a gunny sack and then throw it in the volcanic vent to cook the corn, which the volcanic vent has tons of hypothermophiles. And then they bring the corn home and feed it to their family. So the people are eating hypothermophiles on the corn, and it does not cause them any harm. Of course, the hypothermophiles will not grow in the people, so they're not going to cause harm that way. But it's kind of surprising that, as far as we know, the hypothermophiles do not cause any food spoilage problem. And apparently, they don't make any toxins that would make people sick either. Any question about any of that? All right, let me state that the temperatures here are not rigidly defined, and the range is especially not rigidly defined. There are different species which grow at a different optimal temperature than, let's say, 37 degrees, like Micrococcus luteus grows on the human skin. So its optimal temperature to grow at is 30 degrees Celsius because the human skin is a little cooler than internally in your body. And so Micrococcus luteus grows best at 30 degrees C. We still call Micrococcus luteus a mesophile because it's within the mesophile range as opposed to the psychotrophs. And then, like I said, the range will vary for each species. The minimum temperature it'll grow at, and then the maximum temperature it'll grow at. So the point is, these temperature designations are not ironclad, they're not rigidly defined. Any question about any of that? All right, let's talk a little bit more about food spoilage. Food spoilage usually happens when food is kept between about 15 degrees Celsius to 52 degrees Celsius, and that would be about 60 degrees Fahrenheit to 130 degrees Fahrenheit. This is when bacteria can grow rapidly. And some of these bacteria can produce toxins, so you don't need to have all that much growth. And in 4 hours at this temperature, you should probably throw the food away because the bacteria can grow rapidly enough to make you sick. Now, bacteria can grow slower at a different temperature, meaning from, about 52 to 62. The bacteria can grow slower, and it can grow slower from about 15 degrees Celsius down to, what is that, about 5 degrees Celsius. And so given enough time, the bacteria will grow to a large enough number to make people sick. When we're talking about food spoilage, most of the time food spoilage occurs either because the food is left in this temperature range for too long, or they've refrigerated it in too large of a batch. And so that when the food cools in the refrigerator, it's in this range for 1, 2, 3, about 4 hours. And that's too long. The bacteria can grow rapidly in this temperature range and then cause food spoilage. One example is Bacillus cereus, which has endospores, so it can survive the cooking, and it's on rice, and it survives the cooking of rice. And then if you leave the rice at this temperature range for 4 hours, Bacillus cereus can germinate from the endospore and begin growing, and then it produces a toxin. Anyways, if you keep the rice in too large, or some other food in too large of a vat, it doesn't cool quick enough. You want to have it in a small vat, or split it into 4 different containers, put it in the refrigerator, and then it'll cool quickly, and it'll pass through the danger temperature range quickly enough that the bacteria doesn't have a chance to grow very much. It can grow a little bit, but not very much. And then it doesn't cause food spoilage. Any question about any of that? All right. Let me bring that down here. There are physical requirements, and pH is another one. Most bacteria grow best around pH 7. So between pH 6 and pH 8. Excuse me. Outside of that pH range, many bacteria species cannot grow. That's not all bacteria, but many cannot grow. Trying to remember how low E. coli can grow. And I think it can grow a little bit less than pH 6, but not much less. Like pH 5, E. coli cannot grow. This can be a problem when we're growing things in a lab. The lab cultures tend to, meaning the bacteria tend to digest the media and produce acid byproducts. Those acid byproducts can inhibit the growth of the bacteria. And so to minimize this, we put pH buffers in the culture media. The pH buffer buffers the pH and keeps it from becoming acidic. It also keeps it becoming alkaline, but most bacteria will make the media acidic. Molds and yeast are a little bit of an exception, and they actually prefer growing between pH 5 and 6. So they like an environment that's slightly acidic. Acidophiles, on the other hand, are microbes that grow best in an acid environment, such as in a volcanic environment where the pH can be between pH 0 and pH 3. Remember, your stomach drops down to around pH 1.5 to 2 to pH 3. And it depends what food you have in your stomach and I guess how acidic your stomach is. But if you don't have any buffers in your food, will be an example of that. Something that's low in protein and maybe like watermelon probably doesn't have much buffers. And then the pH will be less. And if you have a lot of protein in your stomach, a lot of other food that buffer the pH, it won't be as acidic. It will be acidic, but not quite as acidic because of the buffering of the food. Anyways, volcanic vents are one place where we have acidophiles growing. These are organisms that prefer growing in an acid environment. Let me end that and make sure I talked about everything here. All right. Another physical requirement is osmotic pressure. Microbes require a range of osmotic pressure, meaning there's an optimal osmotic pressure where the organism grows best. Osmotic pressure is usually due to salt or sugar in the environment, but it can be from any other solute. High osmotic pressure removes necessary water from a cell. So when we put a cell in a hypertonic solution, the cell membrane will pull away from the cell wall, as shown here, and shrink. And this can cause plasmolysis, where the bacteria actually will die. Like E. coli cannot survive long in a hypertonic solution, such as when E. coli falls into molasses, it's going to undergo plasmolysis, where water will leave the cell and that will kill the cell. Osmotic pressure can be used to preserve foods, and humans have been using osmotic pressure for centuries or millennia, and that includes the drying of fruit, where the high sugar content prevents the bacteria from growing, the drying of vegetables, which I'm not sure what, it's a high solute concentration probably. The salting of fish, the salting of meat like beef jerky. What's another example? Molasses, where we have a whole bunch of sugar in there. Something that's salty. Ketchup is a salty food that has high osmotic pressure, but this one is also a sugary food, and both the sugar and the salt help preserve the ketchup, as well as the acid in the ketchup helps preserve the food. And so when we put the ketchup in the refrigerator, I forgot to mention that earlier, only keeps the food good for about a week. But ketchup, if you don't know, can last years in the refrigerator. And that's because it has low temperature, that only gives it about a week. It has high salt, high sugar, it's high osmotic pressure, and then it has a acid environment. And all of those factors help preserve the food. And my bottle of ketchup is at least 3 years old, and it's still good. I should probably get rid of it and get another one, but I don't use ketchup very much, obviously. That's why it's around three years. Any question about any of that? All right. Halophiles are organisms that prefer growing in a high salt concentration. We have two types of halophiles, obligate halophiles and facultative halophiles. An obligate halophile is an organism that requires a high salt concentration to grow. And if you remove it from that high salt concentration, it cannot grow, and oftentimes it will die. A facultative halophile does not require the high salt concentration to grow, but it can tolerate the high salt concentration. And usually a facultative halophile is something that can tolerate greater than 2% sodium chloride, and some can grow in 15% sodium chloride or higher. An example of a facultative halophile is Staphylococcus aureus. It can grow on TSA or nutrient agar or Na, which is around osmotic pressure. So that's 0.9% sodium chloride. So it can grow on that, but it can grow on the human skin, and the human skin can get up to 20% sodium chloride, depending on when you last had your shower or your bath. And that's because when we sweat, we perspire salt along with the perspiration. An obligate halophile, an example would be some organism that grows in the Dead Sea. The Dead Sea is between Jordan and Palestine, and this sea, it has about 20% sodium chloride in the water. Any higher than 20% sodium chloride, the salt precipitates, and that's what's happening in the Dead Sea. The salt actually does precipitate. And so any organism growing there is an obligate halophile. Any question about any of that? All right. In a hypotonic solution, the low osmotic pressure, such as when you're trying to grow something in distilled water, will cause the water to enter the cell by osmosis. The cell membrane will expand. If the cell has no cell wall, the cell membrane will continue to expand as more and more water enters the cell. And that will happen until the cell membrane pops. And that's what happens when you put red blood cells in distilled water, or actually in any water. If the cell has a cell wall, the cell membrane will expand up to the cell wall, and then the cell wall protects the cell, preventing the cell membrane from expanding any further. That will prevent the water from moving into the cell because there's no room for it in the cell. And so the cell wall can prevent osmotic lysis, where the cell will rupture from water entering the cell, causing the cell membrane to burst. Any question about any of that? Most bacteria grow in a hypotonic solution, and they require their cell wall to keep the cell from undergoing osmotic lysis. And this is actually why penicillin G works, because when the bacteria cause an infection in us, the bacteria is in a hypotonic solution, and then the penicillin G damages the cell wall, and then the cell membrane can protrude out of the hole in the cell wall. And then water will just continue to enter the cell. The protrusion will get bigger until the cell membrane pops. And that's how penicillin G kills cells. Any question about any of that? All right. Another chemical requirement is carbon. All cells have to have a carbon source. Half of the dry weight of a cell is actually carbon. Carbon is used as the structural molecule in organic molecules, meaning the backbone. Carbon is also an energy source for cells. Heterotrophs use organic carbon sources. Autotrophs, though, can get their carbon source from CO2 dissolved in the air or the water. Nitrogen, sulfur, and phosphorus are three other elements that all cells require. 14% of the dry weight of a cell is nitrogen. Nitrogen is required in amino acids, in proteins, in DNA, RNA, and ATP. Many organisms obtain their nitrogen source from decomposition of proteins. That is where humans get their nitrogen from. However, although bacteria can get their nitrogen from decomposing proteins, bacteria, at least some of them, can get their nitrogen from another source, such as ammonium ion or nitrate. Green plants, that's their favorite source of nitrogen. Green plants can use ammonia, but they don't like it very much. And you put too much ammonia on the plant and it will kill the plant. And then a few bacteria can actually pull nitrogen down from the air and make it into a biological molecule. And this is called nitrogen fixation. These organisms increase the nitrogen in the area, like if the bacteria were in the cell or in the soil, it would pull nitrogen down from the air and put the nitrogen in the soil. And that actually helps enrich the soil. And if you know about legumes, they have nodules on their roots that have bacteria that can engage in nitrogen fixation. Sulfur is another element required by all living cells. We get most of our sulfur from amino acids or proteins. There are some amino acids that have sulfur in them, but we can also get it from thiamine and biotin. These are two B vitamins, and from DNA and RNA. Many organisms get sulfur from decomposition of proteins, but many bacteria can use sulfate. We cannot, but green plants can. Some bacteria can get sulfur from hydrogen sulfide gas, and that's a toxic substance or a poison for us. All organisms require phosphorus. Usually organisms get phosphorus from phosphate, and this includes humans. We get our phosphorus source from phosphate, like phosphate dissolved in milk. We need phosphorus for making, we need it for DNA, RNA, and ATP, as well as the lipophosphates of the cell membrane or any membrane. Any question about nitrogen, sulfur, or phosphorus? All right, let's move on to trace elements. Trace elements are required by all living things. These are usually inorganic elements required by microbes in small amounts. The organism usually picks up the trace elements directly from their environment. Like humans get their trace elements from the food they eat or the liquids they drink. And these can be enzyme cofactors, such as copper, zinc, iron, elements that we and almost all living life forms require in trace amounts. And we and microbes tend to get these elements just from their environment. Like iron, you get that obviously from eating red meat and some vegetables. I'm trying to remember, it might be broccoli, but I don't remember exactly. You can get iron from some vegetables. Copper, you can get that from eating grapes or especially raisins, probably other foods too. Those are just some that I know. Organic growth factors are required in the media if the cell requires the organic growth factor. Otherwise, the cell would not grow. And that is why Streptococcus pyogenes cannot grow on TCA media. It has to grow on something like blood agar to supply the organic growth factor. And these would be things that the organism themselves cannot synthesize. Does anybody know an organic growth factor that humans require? Anybody? What's an organic growth factor that humans require? If we don't get it, we'll become sick and then eventually die. All right, this is a organic compound that we don't make ourselves. So we have to get it from either the food we eat or the liquids we drink. Examples of organic growth factors include vitamins.
Like iodine?
No, iodine would be a trace element because it is an element.
Vitamin C.
We do require it in trace amounts too. Vitamin C, we do require a certain amount each day. It's a little bit more than trace, but it's small amounts that we do require. Like vitamin C is a organic growth factor, and it's a organic compound, whereas a trace element is actually a element, like iodine, it's an atom. Okay? So that's how you can tell a trace element from an organic growth factor. Other organic growth factors include certain amino acids. We have a name for them, essential amino acids. One example is lysine. We cannot make lysine. We must get it from our diet. And then certain essential fatty acids, we call them essential because we cannot make them. We need to get it from our diet. If the organism requires purines or pyrimidines, then that would be an organic growth factor. Now, humans require either purines or pyrimidines or DNA or RNA or ATP in their diet because we do not make purines or pyrimidines from scratch. We can, however, make them from try something folic acid, try hydrofolic acid, something like that, which is a vitamin. So we can make purines and pyrimidine from THF, trihydrofolic acid. Yeah, it's a folic acid. That's a B vitamin. But we can't make it from something smaller than that folic acid. Any question about any of that? All right. Oxygen can be a chemical requirement. Molecular oxygen is, in a sense, a poison, even for organisms that require oxygen in aerobic respiration. There are certain metabolites of oxygen which are toxic to our cells, and these we call toxic intermediates of oxygen. There are 4 toxic forms of oxygen. The first is singlet oxygen, where the oxygen is boosted to a high energy state. This extremely reactive oxygen molecule, when it's present in your cells, this oxygen will bump into another molecule and then damage that other molecule. Our white blood cells actually make singlet oxygen, and then when they're attacking a pathogen, they will secrete singlet oxygen against the pathogen. And this could be a pathogenic worm or a pathogenic microbe. But white blood cells can secrete singlet oxygen onto the pathogen. And then what they're hoping is the singlet oxygen will damage the cell, the pathogen cell, enough that it'll kill the cell. Any question about any of that? The second toxic form of oxygen is superoxide free radical. This is a free radical of oxygen. It has an extra electron, which is unpaired. So it has -1 charge, but it's unpaired. And whatever molecule this oxygen radical bumps into, it'll steal an electron from. And that can damage the molecule that it's stealing the electron from. If this bumps into DNA, it can mutate the DNA. And even if it doesn't bump into DNA, let's say it bumps into a protein, it will steal the electron from the protein, and then that protein becomes a free radical. That protein can then bump into the DNA and steal an electron from the DNA, and then that can mutate the DNA. So this symbol where, let me blow this up, where we wrote superoxide free radical is O2 with a square O with a negative sign, that refers to an unpaired electron. Unpaired electrons are only found in free radicals. Free radicals are highly unstable. They tend to steal an electron from whatever molecule or atom they bump into. Superoxide free radicals are actually formed during aerobic respiration. It's formed at low numbers, and it's a problem for the cell. So the cell, if the cell is engaging in aerobic respiration, has an enzyme around, superoxide dismutase, abbreviated SOD, SOD, takes two superoxide free radicals and two hydrogen ions to make hydrogen peroxide and oxygen. So SOD neutralizes the superoxide free radicals. Now this molecule here, hydrogen peroxide, is toxic to cells. So the cell has to have something to take care of the product of SOD, or the superoxide free radical reaction, and that is hydrogen peroxide. We have to have something to remove this from the cell because this is toxic to the cell. And that is actually the third toxic form of oxygen, which in general is called the peroxide anion. And this includes hydrogen peroxide, or any peroxide really. And we have two ways of removing peroxide anions, such as hydrogen peroxide. Most cells that live in oxygen have the enzyme catalase, like our cells do. And then we, or the cell, can remove or convert 2 molecules of hydrogen peroxide into 2 molecules of water and 1 molecule of oxygen. And we've talked about this in the catalase test in the lab. The oxygen generates bubbles, and that's how you can tell the cell has catalase when you have hydrogen peroxide around. There are some cells that do not have catalase, and they have the peroxidase enzyme instead, which takes the hydrogen peroxide and 2 hydrogen ions and makes 2 water molecules. Streptococcus and Enterococcus, which do not have catalase, can grow in the air because they have peroxidase to neutralize the production of hydrogen peroxide. Like I said, most organisms that grow in the air, including most microbes and obviously our cells, have the enzyme catalase. Some cells have both catalase and peroxidase, but Streptococcus and Enterococcus only have peroxidase. Any question about any of that? All right, the fourth toxic form of oxygen is a hydroxyl radical. This is the OH with an extra unpaired electron. This hydroxyl radical will bump into another molecule and steal an electron from it. And then that will remove the hydroxyl radical from being a radical, but the other molecule that got the electron stolen from is now a radical, and it'll bump into another molecule and steal an electron from it. And if it bumps into DNA, it can mutate the DNA. You should note that the hydroxyl radical has an extra electron, but it does not have a negative charge. And that's different from the hydroxide ion, which has a -1 charge. And it has 2 electrons there, and so it will not steal an electron. This one has only 1 electron, and it wants 2 to fill the electron shell. If you remember from chemistry, the first shell of electrons only holds 2. And then the shell is filled, and that's what this radical is trying to do when it bumps into it and then steals the electron. Hydroxyl radical is formed by aerobic respiration in trace amounts. It's also formed by ionizing radiation or when hydrogen peroxide reacts with certain metal ions. Another reason why you shouldn't have hydrogen peroxide in your cells. We remove hydroxyl radicals with antioxidants. The antioxidants combine with the hydroxyl radical and then remove it. An antioxidant that actually does that is vitamin C and vitamin E. Unfortunately, that, how should I say, ties up permanently the vitamin C and the vitamin E. So these vitamins, if they're used as an antioxidant, cannot be then later used as a vitamin, meaning that tied up vitamin C, that's tied up to the hydroxyl radical, will be secreted by the body. It will not be used for vitamin C. It's being used as an antioxidant. Any question about any of that? All right. There are chemical requirements of oxygen. Microbes that use oxygen in aerobic respiration, and these we call aerobes, require the presence of oxygen in order to grow and live. They make ATP from aerobic respiration. And then there are microbes that require oxygen to be absent, and these are many, but not all, anaerobes. Microbes can be classified on the basis of the effect oxygen has on their growth. We can grow the microbe in a thioglycolate tube. Thioglycolate is a media that once you take the media out of the autoclave, the media will have no oxygen in it because the media combines with the oxygen, tying it up. However, once you take the media out of the oxygen, the air, and you keep the tube open, meaning uncapped, the air will move into the tube and then create an oxygen gradient in the tube. At the top of the tube, you'll have the full oxygen amount of the air, 20 to 21%. As you go down the tube, you'll have less and less oxygen until you reach the bottom of the tube where you'll have no oxygen. Come on. So at the bottom of the tube is where only obligate anaerobes can grow because they require the absence of oxygen. And as you go up the tube, you'll see most of the growth here. And then you go up the tube, there'll be less growth. Go up the tube, there'll be less growth. Go up the tube, there'll be only one dot now. And that's because there's some oxygen there, not a lot. So every once in a while, our cells can grow, but they're not going to grow very much because there's too much oxygen. So anaerobes, we only see growth in the bottom of the oxygen thioglycolate tube. And as you go up the tube, you'll see less and less growth. For an obligate aerobe, you see most of the growth at the top of the media, because that's where there's 20% or 21% oxygen. And aerobes grow the fastest where oxygen is the highest. As you go down the tube, there will be less and less oxygen. So you'll see less growth, and then right here, only one growth, a little bit of growth because there's a little bit of oxygen in the tube, but not much, and so we won't get much growth. Further down, there's no oxygen, and an obligate aerobe cannot grow. For a facultative anaerobe, these organisms can perform aerobic respiration if oxygen is present. And so we find the most growth in a facultative anaerobe at the top of the tube. And as you go down the tube, you see less growth, but you will see growth all the way down to the bottom of the tube because these organisms prefer aerobic respiration because they can make the most ATP per molecule of glucose. But they can fall back on either anaerobic respiration or fermentation, so they can grow everywhere in the tube. They'll just grow less down here, then up here. Come on, mouse, where there's more oxygen. There's also an aerotolerant anaerobe. These are anaerobes, so they do not require oxygen to grow. But these aerotolerant anaerobes make all the enzymes and I guess metabolites that are needed to neutralize the toxic oxygen intermediates. And so they can grow anywhere in the tube, and their growth will be the same at the top of the tube as at the bottom of the tube. So their growth is uniform. So they can grow in the presence of oxygen, but they don't require oxygen, so they can grow in the absence of oxygen. Any question about the aerotolerant anaerobes? There's also the microaerophiles. These are aerobes, so they do require oxygen, but they require, or they prefer oxygen between 2 and 10% oxygen. They don't grow as well, if at all, at 20 to 21% oxygen. And then less than a certain level of oxygen, there just isn't enough oxygen around, and so they don't have much growth. So the microaerophiles grow best somewhere in the middle of the tube where there is oxygen present, but the oxygen is reduced. A microaerophile is Elobacter pylori, a microorganism that grows in your stomach. Our stomach has about 4% oxygen. And why its oxygen is reduced is that we have some facultative anaerobes as well as obligate aerobes in the stomach, and they use up the oxygen, reducing the oxygen in the stomach. But we can get oxygen into the stomach whenever we swallow, including swallowing air. And so oxygen is brought to the stomach, but it will be less than 20 to 21%. Any question about the effect oxygen has on microbial growth? Let me look ahead. I think that's where I stopped on Tuesday, Thursday, so let me make a note there. Oops, didn't mean to do that. There we go. Let's talk a little bit about culture media. You do know about general media. These are media that many organisms can grow on. We have different types of media. And when we talk about several different types of media, we use the word media with an A, which is plural. When we're talking about one medium, we use the word medium, and this is singular. A medium can be a liquid or a solid solution that contains all of the nutrients needed for microbes to grow. In the lab, we usually would give you a sterile medium, and by sterile we mean it has the medium in it with no living microbes in the tube. And how we get it sterile is we stick it in the autoclave. The inoculum is the microbes that are introduced into the medium. The culture is the microbes growing in or on the culture medium. So the culture is what's grown on the medium. We have a wide variety of media available for growth in the microbiology lab. We have a book about this thick, if you can see this, and it's actually a little bit bigger than this. And on each page of this book, we have one media, which is separate from all the other media on all the different pages in the book. And that tells you how many different media we have available in microbiology. Because I don't know how many pages that would be, but it's at least 300. And that just tells you that we have a lot of different media that can be made in microbiology. Any question about any of that? All right. To support microbial growth, a medium must provide an energy source, a carbon source, excuse me, a source of nitrogen, sulfur, and phosphorus. It must supply trace elements, and if organic growth factors are required, it must supply those as well. Now, if you have a photosynthetic autotroph, you do not need to supply a carbon source because the autotroph can pick up their carbon from the CO2 in the air. So these are only required by the media if the cells require it. And like I said, a photosynthetic autotroph would not need a carbon source. Would a photosynthetic autotroph need an energy source? Assuming you're growing the cells in light? Anyone can answer that? All right, think about that. If you have a photosynthetic autotroph, you would need to supply a sulfur source and a phosphorus source for those microbes to grow. You may need to supply a nitrogen source, unless it's a bacteria that can fix nitrogen from the air, then you need to have a nitrogen source in the media. Trace elements do not usually need to be supplied by the media or the medium because the trace elements usually come from the additives of the media or from the water itself, especially if you're using tap water to make your media. Any question about any of that? All right. Agar is frequently added to the media to solidify a liquid media and make it a solid media. Agar is a complex polysaccharide derived from seaweed. And like I said, it's widely used in microbiology to solidify the media. We could use other agents to make the media solid, like gelatin or starch. However, some microbes can digest gelatin or starch. And if you're having that agent solidify the media, then your media will no longer be solid. Agar cannot be digested by microbes, and that is why we often use agar to solidify the media. Oh, come on. I'm looking for paint here. I'm not seeing it. I must be in the wrong thing. Yep, I was in the wrong thing. When we grow media in a broth culture, we usually grow it in a tube. And that's because if you grew a broth, put a broth in a petri dish, it'd be too easy to spill the broth and contaminate the culture. So in a petri dish, we usually have a solid media. In a tube, we could also have a solid media, and we have two types: a deep, which has the agar going across, straight across, and then a slant, where the agar is put in a slant, kind of like that. I'm not drawing a very straight line. You could also have the slant, something like that. It's going like this, and then you put the slant like that. And let me erase that right here. So it's growing like that. Both of these are slants. Any question about that? All right. If there's no questions about that, I'm going to stop here. And I think in the lab, I'll be there for the first 15 minutes to answer questions. However, after 15 minutes and if there's no further questions, I'm going to log out. We have no lab today. All right, any questions? If not, I'll see you later.